So good evening everyone. Can you all hear me at the back? Okay, good. Before I start, can I make sure that you can all see one of the handouts? It's going to be audience participation later on, so make sure you can see it. So I work at King's College London, and the one question that my lab would really love to answer is, what does the eye tell the brain? And we'll often get asked by funding agencies, people, members of the public, even scientists outside other disciplines, is why is that question important? And the definition of importance really varies according to who you're talking to. So for some, the question is important if answering it helps you understand a disease or solve a particular problem. For others, the question is important if it addresses something fundamental and fascinating. And actually the two are not mutually exclusive. There are many examples of people studying basic, simple biology, and given time, the answers that they've come up with have had a profound influence on how we understand the brain, profound influence on how we cure diseases. But we're basic scientists. We really want to understand how the visual system works. And how the visual system works is not intuitive. In fact, it's actually quite strange. If nothing else, I hope this short talk will lead you to question your own version of reality. So I think I'll start just by describing the piece of neural tissue that we're interested in. So this is the retina. So the retina is the piece of neural tissue in your eye that sends information about the visual world to the brain. And it sits at the back of your eye. It's only about 1 millimeter thick. What it does is pretty amazing considering how thin it is. And photons of light are focused onto your retina via the action of your cornea. This is a static structure on the surface of the eye, and the lens, which can change shape. And this allows you to focus on near and far subjects. And if you were to take that retina out and cut it in half, and then look at the cut surface under a microscope, you'd see it's divided into 3 layers of cells. And in the first layer, the outer layer, are the photoreceptors. And these are the cells that convert photons of light into the very first neural signals in the visual pathway. And in our retinas, we have 4 types of photoreceptor. We have the rod photoreceptor. So these are really sensitive, allow us to see at night time. And we also have 3 types of cone photoreceptor that are sensitive to different wavelengths of light. You have the red, the green, and the blue cones. And these allow us to see during daylight hours. Those photoreceptors will send their signals onto cells in the second layer of the retina. And there are broadly 3 different types of cell in this layer. You have the amacrine cells, the bipolar cells, and the horizontal cells. And there are hundreds of different types of these different cells in the second layer. It doesn't matter what they do for now, but for now, just accept the fact that those cells are assembled into really complex circuitry that does something amazing, and we don't know how it does it. But anyway, these cells in the second layer pass their signals onto cells in the third and final layer of the retina. And this layer contains the retinal ganglion cells. These are the cells that we study because these are the cells that tell your brain what's going on in the visual world. So the question is, okay, what are all these different cell layers in the retina actually doing? And another way of phrasing that question is, okay, how is information about the visual world represented by the activity of cells in those different layers? So what you would do as a neuroscientist, and this is a really common experiment, it's repeated throughout sensory systems, is to record from a single cell in the particular layer that you're interested in. And then you show that cell something. You probe it with a stimulus and say, how does that cell respond? So let's imagine we do that experiment on a single photoreceptor cell. We're recording from it, and we're going to probe its functional properties using a very small pinpoint of light. And you move that spot of light around, chances are that photoreceptor will do absolutely nothing. That is until that spot of light falls on a very small and particular region of visual space. You move the spot of light away from that region, the photoreceptor will stop responding. So that tells you that each individual photoreceptor is sensitive to only a very small part of visual space. Now if we hold that spot of light still and just change the intensity of it, you'll see a corresponding change in the output of the photoreceptor. So that tells you that an individual photoreceptor is recording light levels at each point in visual space. In our retinas, we have about 130 million photoreceptors, and you can think of these as a whole array of spot detectors measuring light levels at every single point in visual space, exactly like the sensor in a digital camera. So do we have our answer? Is that what the retina is telling the brain? You have this pixel-like representation of the visual world at the level of the photoreceptors. Is this continuously being streamed to the brain, which then assembles those pixels into a coherent image? And the answer is no. And the reason the answer is no is actually quite simple. If that's how the visual system works, if that's how the retina was talking to the brain, why would you need all that complex circuitry that sits downstream of the photoreceptors? That circuitry in the retina must be doing something to those photoreceptor signals. The other reason we know that this isn't the case is I said that you've got 130 million photoreceptors in your eye, but you've only got 1 million retinal ganglion cells. Remember, these are the cells that connect the eye to the brain. So that tells you right away that it cannot be the case that every single photoreceptor has its own dedicated ganglion cell telling the brain what that photoreceptor is doing. So the obvious question then is, what are those ganglion cells telling the brain? So to address this question, you need to do your experiment again. You record from a single photoreceptor, you show it the pinpoint of light that was so good at getting that single photoreceptor to respond, and then just see what happens. Chances are, in fact, be sure of this, that that ganglion cell wouldn't give a damn. It doesn't care about pinpoints of light. So like any good scientist, you blame your recording equipment. You turn it on and off, you whack it, you phone up the manufacturer and complain. Turns out you're recording, there's nothing wrong with your recording equipment. So you have to redesign the experiment. Say, okay, let's try a different stimulus. So, okay, let's start moving the spot. And now your ganglion cell crackles into life. And you think, oh, we've solved it. Ganglion cells are telling the brain about things in motion. So, like any good scientist, you repeat the experiment. You go to another ganglion cell, record from it, show it the spot of light. Lo and behold, it does nothing. So you redesign the stimulus. You show it something else. You show it a bar moving across the visual field, and now the cell crackles into life, but only if the bar is of a particular orientation. So you keep repeating these sorts of experiments, you keep doing them, and you come to two conclusions. The first is that these retinal ganglion cells are not telling the brain about spots of light. We already know that. What they are telling the brain about is about particular features in the visual environment, such as edges of a particular orientation, or things in motion, or flicker. The other conclusion that you come to is that there are many different types of ganglion cell, and each type of ganglion cell is telling the brain about these particular features, such as motion or edges of a particular orientation. So what your retina is doing is converting this pixel-like representation at the level of photoreceptors into these parallel streams of information. And each stream is telling the brain about a particular feature in the visual environment. The brain does something pretty remarkable. It assembles all these disparate pieces of information, puts them together to build a coherent internal picture of the world. It's a pretty remarkable thing that the brain does. So the question you ought to ask yourself is, why does the retina bother doing this? You have a perfectly good representation of the visual world at the level of the photoreceptors. Why bother changing it into these parallel streams? And there's two reasons that we think this is the case. The first is, the world is not made up of pinpoints of light. It's made up of edges of a particular orientation. It's made of shapes and blobs of colour and things in motion. So it actually makes sense that the retina should be telling the brain about things that actually exist in the visual world. The other main reason concerns one of data compression. So I've told you we have 130 million photoreceptors, but only 1 million retinal ganglion cells. So there's a real compression in the amount of data that your brain has to deal with. But despite that, over 50% of your brain is already devoted to vision. Can you imagine how big your brain would have to be if it now had to deal with 130 million ganglion cells rather than 1 million?
Sorry, what was the number of percentage you gave? How
So
much of the percentage?
50%. Roughly 50% of your brain is devoted to vision. which, by the way, is why we find it so easy. We make no effort to see. If 50% of your brain were devoted to playing chess, you'd all be grandmasters. So let's do a simple experiment. Okay, how big would your brain need to be if you did have 130 million retinal ganglion cells? Well, as it is, your brain weighs about 1.5 kilograms. If you had to deal with that extra information, your brain would weigh about 100 kilograms. That's like carrying the weight of an England rugby player around inside your head. Not something I'd wish on anyone. So the retina has evolved as a filter. It filters the visual information and streams it to the brain. And this is really efficient, but it's not without consequence. And the consequence of that is that your brain is getting certainly fragmentary and incomplete information about the visual world from the retina. And so what your brain has to do, to a certain extent, is interpret the information that it gets from the retina. It has to make certain assumptions about what's going on out there. And I choose the word interpret very deliberately because it implies, I think, 3 different things. So firstly, it implies that there's opportunity for misinterpretation, that the model you construct inside your head is actually incorrect. The second thing it implies is that there's opportunity for alternative interpretations, that what you see is not necessarily what the person sitting next to you sees. In fact, your own interpretation of the visual world can change over time. And the third thing is how you interpret anything depends on what you know and your experience of how the world actually works. So that's to say what the visual brain is doing is using new visual information and comparing that with stored information, i.e. knowledge and experience. And it's using that knowledge and experience to validate and test the information that it's getting from the retina. So these are all fairly abstract concepts, which is why I've given you those pictures. So those pictures are optical illusions that demonstrate that all 3 of those things are happening. So if I could just get you to look at the first picture, which is the picture, it's a cartoon picture of the 2 tables. So we all agree they're tables, right? Okay, so the table on the left is square and the table on the right is a rectangle. Does anyone disagree? So we all agree on that. Everybody is wrong. The tabletops are exactly the same size and shape. And if you don't believe me, grab a pen, trace round one, and it will perfectly superimpose on the other. Even when I've told you that, you can't see it.
What about we can understand the areas, the total areas of each, not the actual shape.
The shape is exactly the same. So if you were to draw around that and superimpose it on that. So what does this reveal about our visual systems? Well, what it's revealing is that the model you've created, and you've created the most plausible 3-dimensional model from 2-dimensional information, and the model you've constructed is wrong. So your visual system is certainly imperfect in this case. So now we're going to move on to the second picture. Many of you would have seen this already. It's a picture of a woman. We all agree on that. But I can guarantee you won't agree on the age of the woman. About half of you will think it's a young woman. The other half will think it's an old woman. So hands up who think it's young. Okay, hands up who think it's old. There you go. So that's telling you right away that we're all building our own separate interpretations of what's out there in the world. And it's not the picture it's changing. The picture can't be changing. It's your interpretation of it. The other thing this demonstrates is your visual system is very active. You don't just simply download an image and store it. Your brain is constantly re-evaluating the information it gets from the retina and constantly generating these models. And in this case, your interpretation can change over time. And the last image gives you an example of how you use visual information and combine it with experience. So there's clearly something strange about this picture. There's a very large man and an implausibly small woman. You know it's not correct, but the only explanation, unless you've seen this before, the only explanation is that he's very large and she's very small. Actually, the reason they look that way is because the room is a weird shape. He's actually much closer to you and his corner of the floor is much higher, whereas she's further away from you and the corner is much lower. But what you see are two people of completely different sizes. What you don't assume is that the room must be a weird shape. And the reason you don't assume that is because all rooms are roughly the same shape. So this is an example of how you're using visual information and using past experience of how the world actually is to construct a model. And in this case, you're getting it wrong again. So I hope I've given you some insight into how mysterious and fascinating the brain is. I haven't told you anything about what we do in the lab. If you would like to know, I'll tell you over a bit. Okay, thanks very much.